Hi, and welcome to the Origins Podcast. I'm your host, Lawrence Krauss. In this episode, I had the long-awaited pleasure, finally, of having Jennifer Doudna on the program. Jennifer won the Nobel Prize, along with Emmanuelle Charpentier, for discovering CRISPR, a technique, a gene editing technique that's revolutionizing biology and offers the promise of being able to edit the human genome in ways that can cure disease and potentially do some things that might sound dangerous, like create designer babies. It's an important tool, and it's changing not just biology, but the way we may think about ourselves as human beings. And it's therefore important for society. And in order to understand it, it's important to understand the science. So what I wanted to do with Jennifer was talk about her own experience of discovery, which was based on curiosity-driven research and not an effort to actually solve the problem of how to edit the human genome, but also involved serendipity, many aspects of science that are really important. So I wanted to use her discoveries in microcosm to understand science, and then to discuss the background behind the important questions, the important ethical questions that CRISPR raises. And we had a wonderful discussion of an explanation of biology, of CRISPR, and her own experiences both in the lab and outside the lab. It was fascinating for me. I knew it would be. It was a wonderful discussion, and she was great. And I think you'll find this educational and also thought-provoking, because CRISPR opens up the possibility of changing what it means to be human. And the Origins Podcast is all about bringing you closer to these discoveries that affect the challenge, that produce the challenges of the 21st century. And the only way we as a society can address those challenges is by understanding and communicating the fundamental science. And this, in my mind, was just a perfect example of that. And I hope you enjoy it as much as I did. You can watch it ad-free on our Substack site, Critical Mass, or you can, and if you subscribe to that site, you'll support the Origins Project Foundation, our nonprofit foundation that produces the podcast, or you can watch it on our YouTube channel. And if you do, I hope you'll subscribe to our YouTube channel, or you can listen to it on any podcast listening site. No matter how you watch or listen to it, I think you'll find this discussion with Jennifer Doudna, this great scientist, talking about a great discovery, remarkable as I did. So with no further ado, Jennifer Doudna. Well, Jennifer Doudna, thank you so much for coming on. I've wanted to have you on for a long time. Obviously admired your work, and it's really nice to connect, if at least virtually. So thanks for being here.
It's a pleasure to be here.
And I want to, and we'll go a deep dive into the remarkable science that you've been involved in and its implications. But this is the Origins Podcast, and what I like to do is really begin by finding out how people got to where they are, to the point of why we're talking about what they're doing. And I do know some aspects of your childhood, which you talk about in the book you wrote, The Crack in Creation, and some other research I've done. But I wanna go into it a little further. Clearly, it's clear that your father had a big influence on you. That you mentioned two things that I can remember. One, giving you Jim Watson's book when you were 12, I think. And the other was connecting you. He's a professor of, what was he a professor of again?
American literature.
American literature. And in spite of being a professor of American literature, he didn't seem to mind you were going to science. But when you were a fresh person, he got you a summer job in a lab, and that hooked you up. So he knew given the Watson's book, he must have known you liked science. Where did your interest in science get kindled? Was it through discussions with him, or anything else, or reading, or TV?
Well, I grew up in an interesting place. I grew up in Hawaii, but not in fancy Hawaii. I grew up on the Big Island in a town called Hilo. It's very rainy. It's very beautiful. It's kind of a working class town. It has a branch of the University of Hawaii campus on it. And that's why my father was there, because he was a professor at that branch campus. And I had the opportunity to experience that culture, to explore the island and all of the fascinating biology there. But I also My father was a professor of literature, and so our household was quite intellectual in the sense
Yeah.
that there were always discussions going on about all kinds of topics. My dad was an avid reader; he read everything, as far as I could tell. He just there wasn't really a topic he wasn't interested in, and so he was always reading things, and he would often, toss books my way. And I think it was partly just to see what took. And so with the Jim Watson book, it actually took me a little time to pick it up, I'm embarrassed to say, but once I
No.
did, I couldn't put it down, and I
Yeah.
was really fascinated, and I think that was something that, my father noticed and realized that, I had a real interest in science.
Now, w clearly, he was a big reader. Were you a big reader too? Did you like to read everything? I noticed you first thought Jim Watson's book was a mystery story, which it sort of is. Did you read literature primarily, or did you read popular science other than that book, or
Yeah, I
more?
did read I did read a lot. I did, everybody has to recognize this was, before the internet. It was
Yeah,
before TikTok,
yeah,
it was before any of the when I tell my son this, he's shocked. The world existed before those things, but yeah, and being in a small town, we didn't have a lot of entertainment. So, yeah, I did a lot of reading. I read, yeah, pretty much everything. I read westerns, I read histories, I read a lot of fiction. I read young adult fiction. I didn't To be honest, I did not read a lot of science fiction, but when I did read science fiction, I read the classics. I read Arthur C. Clarke
oh, yeah.
and Ursula Le Guin, and
Yeah,
I read things like that. So I really enjoyed stories and nonfiction that featured people doing interesting things.
absolutely. I wrote once one of my books is called The Physics of Star Trek. I wrote many years ago, which became very popular, but everyone thinks I'm therefore fascinated with science fiction, but I used to read it when I was a kid, but until I got to the point where I realized that science was much more fascinating than science fiction, and then I switched over. But so I know about your father, but what about your mother? I don't know much about her influence and what she did. So why don't you tell me?
She was a, lecturer in Asian history, so she was a history buff,
Mhm.
and, she was, originally, kind of a homemaker, I would say, in our when we were really little kids, but, after we moved to Hawaii, she started teaching at the local community college, and she got fascinated with Asian history. And so she ended up going back to school in her 40s, and she
Oh,
got a
great.
degree in Asian, history. She got a teaching degree, and then, that's what she taught for the rest of her career.
So interesting. She wasn't one of the one. I mean, if she went back to her school in the 40s, wasn't one of those things that where she graduated around the same time you did or anything like that. That were you and your
Actually, it probably was, actually. Yeah, I think it was. I think I had already left home. I think I was maybe in college, and, yeah, we probably did graduate around the same time. Interesting.
interesting.
Yeah. Yeah.
It probably meant a lot to her that you did. Um, so, um, as you say, you didn't read science fiction, you read science books, but now I want to ask the important question, and I always have to ask people: why biology and not physics?
It's a great question. Actually, it's a great question. I think well, it actually I have to tell you that initially it was neither. It was chemistry. I
Chemistry.
was really interested in chemistry. I had a great chemistry teacher in high school, and
That is such a rare thing because if you look at high schools, among the worst teachers, it always seems to be chemistry teachers.
chemistry?
Yeah. Yeah.
No, ours was great. Miss Wong was her
Oh,
name,
oh,
and
great.
she was someone who really emphasized discovery and experiments. And it was the first time that I had this concept that science was not about memorizing a bunch of facts.
Yeah.
It was actually about figuring things out. And I always loved solving puzzles. That was another thing we used to do in our house a lot was we always had a jigsaw puzzle going. We were always working on crosswords and, solving other kinds of word game kinds of things. And so, for me, realizing that science was figuring out the answer to puzzles
Yeah.
was
Solving
great.
puzzles.
Yeah.
Yeah, no, and it's interesting you say it wasn't memorizing, it's the first time. I wanna ask you about this because, um, uh, uh, a, uh, Leon Letterman, who was a friend and colleague of mine, Nobel Prize winning physicist, and I have worked a lot to try and change the way science is taught? Because schools tend to teach science, biology first, then chemistry, and then physics. And we argued it's the exact wrong order because physics is the basis of chemistry, chemistry is the basis of biology. And
Well,
we know the reason they do it. I mean, biology seems friendlier and touchy-feely stuff, but it does give you the impression because you get these concepts that you're just told exist and that you sort of have to memorize it. Whereas if you learned it the other way around, you'd realize, where it came from. Have you thought about that much at all? Or
I think you have a good point there, honestly. It actually does make a lot more sense to teach it in that order, but somehow they don't. And actually, that was true in college too. In
yeah, but
college, it was the same thing. We tended to, study the more touchy-feely science first and then get into the hard stuff later.
yeah, I mean, obviously the reason is they somehow think math is the scary part, and there's not as much math in biology at the beginning, and unfortunately though, because the way things work now, a lot of because it's taught in that order, people, they take their electives, and so they take their first science, they have to take biology. Most of them never get around, maybe even to chemistry or much less physics at all.
It's true. Yeah, I know, it's too bad.
Now, so that's interesting. I think the chemistry. Well, you became a biochemist, so you chemistry remained your primary interest. So did you you got turned on to chemistry when you went to college? Did you know you wanted to do chemistry first, or did you? How
Actually,
did it?
I knew I wanted to do biochemistry.
You did
Yeah, because in
because of
high
Watson,
school,
or
in high school, I had just, yeah, I love chemistry, but I realized that what I was really excited about was learning about the chemistry of life. And
yeah,
that did kind of start with the Watson book, but it continued to thinking about the chemistry of cells and all kinds of questions that come up when you start to really dig in. And we also
absolutely.
had a series of scientists that came in from around the state of Hawaii to talk to our high school class. And one of them was a biochemist who explained that she was studying how cancer cells grow and why normal cells become cancer. And I thought that's so interesting. I want to work on that.
Okay, that's great. No, yeah, well, that's I mean, and getting exposure to things like that is really important. There are many things I'm not sure I regret, but my mother wanted me to be a doctor, and for many reasons, my brother a lawyer, he became a lawyer, but she told me doctors were scientists, and I got interested in science, and then I discovered in high school that maybe doctors weren't scientists. But the big point was I dropped biology because when I took it, and I'm a little bit older than you. Um, uh, Maybe a decade or so. Um, it was where I was taught, it was just memorizing. It was just like memorizing the parts of a
Wow.
frog, and because the chemistry of life hadn't yet permeated. This was in the '60s, and that explosion, that fascination, which I've had to learn after the fact, wasn't there. But I can, it's just amazing. I mean, it's just amazing how things change, and it's also satisfying because in a way, because the fields of like physics and biology have merged in many ways, because in so many ways, and they're not so different, but they used to be. So you knew you wanted to biochemistry. You went to Pomona. Is that just because you wanted to stay in California, or you're in Hawaii because you didn't want to go too far east, or was it a liberal arts college aspect?
Well,
Or
my parents really wanted me to go to a liberal arts college. They were both Oberlin College
oh, okay.
graduates.
I
And I
taught
thought,
it. I was the
"Ohio?
chair.
Oh, no, I
Yeah,
couldn't
I know.
see
I was the chair of the physics department at Case Western for a while, so
it."
yeah, so I know what
Yeah.
that.
I mean, it's a great school, but I
But
thought, it's
Ohio, yeah, I
a little
know.
far from Hawaii and a little bit cold in the wintertime, a little flat. So I ended up, looking for opportunities elsewhere, and we had family friends who had also attended, Pomona, and they told me that Pomona had a wonderful science program. So I looked into it, and actually it was very interesting because Pomona at that time, this was in the early '80s, they had the only one of the only undergraduate biochemistry
Major.
professors
Oh,
that I could find at a college like that. It was a relatively new discipline, and
yeah,
so
sure.
most colleges had, people who were strictly biologists or chemists, but not somebody doing both. So that was very exciting for me.
Oh, yeah, it's really important, and kids often ask me where to go, and generally I say that, it doesn't really matter where you go. You can get a great education anywhere. Get and go to a place where you're challenged by other students if you can find a place like that, and generally get to know professors. And if you do well, you can go to the best graduate schools. I mean, I went to a small college, and then I went to MIT. You went to a small college, then you went to Harvard. Um, and it's a great way to get excited is if you can work with a professor. Now, you worked that summer in a lab in Hawaii that your father got you hooked up to, and that was the first time you sort of got that joy of discovery, which we'll talk about it later, I think. But were you able to work in labs as an undergraduate, at Pomona?
I certainly did. Yeah, I did from the very beginning. I worked in several different labs. I did some interesting things. I washed dishes.
Yeah, of course.
That wasn't so interesting. Actually, it was kind of fun. I washed dishes, and I also worked in an orchid lab, a lab that was studying how viral infections occur in orchids and how to prevent them. I worked in a lab that was studying how muscle cells contract, and we were working in mice. So we were
No.
working in mouse tissue. And then I landed in my biochemistry professor's lab where I was studying soil bacteria. So quite a range.
Oh, yeah, no, that's quite a range. Now what I mean, other than reputation, did you choose Harvard because I mean, did you know about Jack Shostak, or did you just choose it because of the department and okay, and
Yeah, and my undergraduate professors told me that, they thought that program was really strong. It was interesting because I didn't apply very many places. I think I only applied to 3 or 4 graduate schools, which, nowadays sounds kind of crazy, but
yeah.
I couldn't, you had to pay, I don't know,
Yeah.
60, $70 per application. I didn't have a lot of money. My parents didn't have
Mm,
much, and so I couldn't afford to apply to 10 or 15
yeah,
or 20
yeah.
places. And furthermore, I guess I sort of imagined that I might end up at my father's alma mater, which was the University of Michigan at Ann Arbor.
Oh, okay.
So I applied there. I also applied to Penn,
Mhm.
and then kind of on a lark, thinking I'll never get in, but what the
Yeah.
heck? I threw in an application at Harvard. I was very surprised when I got accepted.
Yeah, no, yeah, well, I grew up in Canada, I didn't know about the American system, I also only applied to a few, and, and, yeah, and didn't get into any except for MIT, but that was okay.
Not too shabby.
Yeah, it was okay, it worked out. Um, okay, uh, through serendipity, or which is a story of many things in that we'll get to in terms of the science you discovered. The term serendipity just kept coming up in my mind as I was reading about your work. Um, you ended up working with Jack Shostak, which turned out to be was that again? Did you choose that area, or was it just one? I mean, I know what it's like for graduate students. Sometimes you just end up in and you luck out. But were you beginning to focus? Jack had already moved to the origin of life at that point, or had he? Or
Actually, he was kind of in transition. So he was known to all of us graduate students as a kind of a young prodigy type, professor. He's very young, he's very smart, doing really interesting work. He was at that time working on how DNA molecules in cells recombine because we know that, when cells divide, and this is also, of course, very important for inheritance, chromosomes will recombine and, they kind of make up an amalgam of, genes from mom and dad. And
Mhm.
and then that's how traits get passed along, and how the species is able to ensure that there's enough diversity that you can avoid, deleterious mutations and things
Mhm.
like that. So he was fascinated by those kinds of questions. And he was studying this process in yeast, so in a fungus, yeast cells, actually using the kind of yeast that bakers and brewers use. And so I went to his laboratory to learn a little bit about working with yeast, which I had never done before. And when I got there, he said, "Actually, I've gotten very interested in the origin of life. And I want to start researching this, and I have some ideas. And would you like to be the first student to move in this new direction?" And I
Mhm.
said, "Sure." And it
Great.
was great because, he was very, very excited about this. He was reading a lot about it. It was an exciting time in the field when there were some new discoveries coming along that we could act on. So it was a great decision, but it was quite serendipitous, to be honest.
Yeah, no, and was the yeast were the experiments on yeast, is that what he won the Nobel Prize for, later on?
Well, not exactly. Let's see. Not exactly. He actually, but it was sort of related in a way because he what he won the Nobel Prize for was for how the ends of chromosomes
Yeah.
are maintained.
Mhm.
And it turns out that in yeast, the chromosomes are somewhat unique, but he was studying how chromosomes are maintained in other kinds of organisms that are a little bit more similar to the way human
Oh,
chromosomes are
okay.
copied. And that was the work that was recognized for his Nobel Prize.
Now, he'd gotten interested in the origin of life. At that point, had, uh, Czech and Altman already sort of discovered what led to the RNA world, that RNA is both a work that you later got involved in, essentially, that RNA is both a genetic coder and also an enzyme, or at least a catalyst.
Yeah, they had just published that work when I arrived in Shostak's lab, and in fact, this is what Shostak was so excited about, like, sort of pursuing some ideas that came out of that because it was a time in the field when it was very new to think about the way that a certain kind of molecule, in this case RNA, could have been the primordial molecule that allowed life to get started, and this
Yes.
was the idea that he really wanted to explore.
Yeah, no, that's when focus went from DNA, which had been, since Watson and Crick had been the big thing, to suddenly RNA becoming of interest, and RNA in one way or another has been a great focus of your career, and we'll go into that. I wanna, we're gonna do some biology lessons, so I want everyone to be up on the same page before we talk about CRISPR and then the consequences, and we'll try and do it in less than a full term, but uh, I was intrigued. It's probably worth mentioning. You start the book in a sense talking about this case, Kim, who had a disease and basically was spontaneously cured. You want to just talk about that for a second or a minute?
Yeah, I mean, I think what's fascinating is that when people have studied genetic diseases, it's often been through investigating rare cases that we've made fundamental discoveries, and that includes finding genes that are causal for disease, and there are probably about 7,000 genes that or diseases in humans, where
Mhm.
there's kind of a clear
Yeah.
known single genetic basis for it. And then occasionally there are spontaneous cures that happen because of sometimes this process that we just mentioned, DNA recombination, where DNA molecules will recombine in such a way that the mutated gene is corrected or removed.
Just amazing.
And it's those kind of, originally before it was possible to do anything fancy in human cells, you could observe these cases rarely in the human population and draw inferences about what genetics were for a particular disease.
And interesting, and it's basically, it's the story of control versus evolution, in the sense that yeah, by recombination, by accident, you might cure something, and the idea is, can we can we learn from that and maybe make it happen in a controlled way, which is really what
Exactly.
your CRISPR discovery allows us to do. Um, Now, let's just talk biology. So, okay, DNA, 4 base pairs, and they can connect up GC and AT, and that creates this double helix. Okay, but I just want you to explain some terms which maybe we'll use later. Which is, first of all, transcription.
Well, that means making a copy of a gene, which is originally in the form of DNA, and copying it into a molecule of RNA.
Okay, and RNA is like DNA, except two of the base pairs are different, but it can hook up to DNA and transmit that genetic information because it has the same uh, series
Kind
of
of
letters.
chemical
Yeah.
structure, Mhm.
Okay, translation.
Translation means making a protein. And the way that that happens in biology is that there's a code that is originally in genes in DNA That code is copied into an RNA molecule, as we just said, by transcription. And then that RNA can be translated, meaning that there's a machinery in the cell that will read the genetic code in the RNA and link together the components necessary to make a particular protein. So proteins, probably people know, or maybe they do, are made of amino acids. And so what the code does is it tells the machinery which order and which amino acids to hook together to make a particular kind of protein. It's a very incredible process,
It
actually.
is really an
Yeah.
incredible process. You'd almost think there was intelligent design if you didn't know better, but, which we do. Uh, it is amazing. These codons, 3 letters turn into an amino instruction making amino acids, and then one uses one of these, what, 21, 20, how many amino acids are used? 21?
Yeah, about 20,
Yeah, 20.
maybe 21, rare, a couple of rare ones.
Okay,
Yeah.
and then just since we'll talk about the human genome.
Yeah, what you're asking me,
I
what
mean
is
I
it?
mean, I'm
What is the
asking you to
human?
define these terms just so
Yeah.
I wanna put everyone on the same wavelength because,
Yeah. No, it's
I think
important.
it's important.
It's very important. So, what is the human genome? The human genome is the entirety of DNA that's found in a human cell. So, it's the code of life. It's the a set of instructions that tell cells how to develop into a human being, meaning making our brains and our hearts and our lungs and our, all our other tissues. And then not only that, but functioning together as an organism. It's again, it's truly incredible that a molecule like that could contain the instructions to build such a thing.
Yeah, it's remarkable indeed, um, and okay, mitochondrial DNA, which turns out to be important later on.
Right. Well, people might have heard of mitochondria because sometimes you see that mentioned in the news because of things like aging and what happens to our mitochondria when we exercise and things like that. And that's because mitochondria are the little energy factories found in cells. So they are responsible for taking chemical energy in the form of food that we eat, so sugar molecules in particular, that go into the mitochondrion and they're converted there by a chemical process that turns those breaks down the sugar molecules into much simpler molecules and allows things like our muscles to function and our brains to think and all of those good things to happen.
But the mitochondrial DNA, which the fact that it comes from the mother, I just wanted
Oh,
to.
right. Yeah, no, of course. I mean, the DNA is interesting because like you said, yeah, it's inherited through the mom and it's a very small instruction set compared to what we find in the rest of the cell. It's an instruction set that is geared mostly towards what we just said, kind of energy harvesting, energy conversion. And because it's such an important part of the cell, these mitochondria, when somebody inherits a defective gene from mom that is a mitochondrial gene, that can cause disease. So we know there are a number of diseases that are inherited in that fashion.
Yeah, I think it's important when we get later on that, yeah, normally we think of 50% of our genes come from mom, 50% of our genes from dad, and that's in the chromosomes, but there's this little extra bit that just comes from mom in the mitochondria. And mitochondria also, I guess, are important because they're a great example of evolution, right? Uh, they, I guess since the work of Lynn Margolis or maybe others that that they probably got this function to be able to handle oxygen and generate power by eating another, basically a symbiotic relationship of eventually combining two entities and
Two
making one.
kinds of cells, yeah. They probably came from bacteria
Yeah,
originally.
yeah.
Yeah, yeah.
Um, okay, last two things. Recessive genes.
Those are the genes that can be, superseded if you have kind of a good copy of the gene. So just to put that into context, so, all of us have in all of our cells except for our egg and sperm cells, we have 2 copies of every gene. And so you could imagine a situation where somebody inherits a defective copy of a gene from one parent and a quote normal copy from the other parent. Well, if that gene is recessive, as you just said, then it might not actually matter for the person in terms of their health, right? It's kind of almost like it's hiding. It's
Yeah.
hiding in the cells. It's and you don't notice it. You only might notice it if that person goes on to marry someone or have children with somebody who also has a defective copy of the gene and their offspring inherit those two defective copies, right? Then you'd have two copies and you would observe an effect. So it's it's kind of it's like a little bit like a Trojan horse in a way.
Yeah. It's there, but it doesn't always manifest it. And then
Right.
dominant inheritance is the opposite, right?
The
It's
opposite, right? You have one bad copy, you're in trouble.
Yeah, it'll always take over.
Yeah.
Okay, I think that's the tools we need probably for the rest. Now, viruses, um, part of the fascinating parts of this story is that what is now a tool that's changed biology and changes and may change the way we are as human beings, began thinking about viruses and bacteria, which is wonderful. Um, what do viruses do? And also, I want to ask you a question. Uh, Do you think viruses are alive? I mean, I
No.
I know they're not, but you know what? I I guess the question is, what's life? And one of the chapters in my last book is about that. I mean, they don't have the machinery to reproduce on their own, but they can hijack the machinery of a cell to reproduce. And it seems to me that's pretty almost like life.
They can, and, you bring up an interesting point because I think what's happened is that as we study viruses and cells more and more, and this is a lot of the work of some of my colleagues here at the Innovative Genomics Institute are working on this kind of thing. We find that, the lines are blurred in a way, right? So you can find, uh, DNA that seems to belong to a virus, and yet it seems to also encode most of the machinery, but maybe not quite all,
Mm.
that you would need to be self-reproducing, and vice versa. There are cells that are very, very small and they're very streamlined. They're almost virus-like, except that they do seem to have all of the necessary components for reproduction. So it's a bit of a blurry line. But you're right. I mean, people have been fascinated by viruses ever since we've had biologists because they are able to take over cells and hijack their machinery to produce more viruses.
Yeah, no, I mean, it seems to me it's almost like cuckoo birds. I mean, it seems to me like all they are is a kind of life that's learned how to be more efficient. Why bother having all that extra baggage when you can
Wow.
just find a handy cell to do it for you?
It's a great solution. Yeah.
Yeah, it's a great solution for life, and I wonder, well, in terms of thinking about life's evolution, they obviously play an important role. Now, um, viruses obviously edit genes because they go into a cell and they cause it to make more viruses, and there are two types of recombination after uh, long sequences of DNA they get cut or moved around. There's homologous and illegitimate. I want you to tell me about the two of them.
Um, well, let's start with homologous.
Okay.
So this means that you have two segments of DNA that are the same, and so they can overlap with each other, and they can allow an exchange reaction to occur that allows one DNA strand to swap into the other or kind of replace the other. And so that's the type of DNA recombination we call it that happens a lot during, for example, after an egg cell is fertilized, and you have cells that are dividing very quickly. Those cells are growing, and their chromosomes are recombining, and that type of recombination that we just talked about is typically what's going on.
Whereas illegitimate recombination is
Is where we have a situation where maybe there's often just a very, very small section of DNA that is similar in letter sequence to another piece of DNA, but it leads to a recombination event in any case that maybe shouldn't have really happened, but does. Um, and this is the kind of thing that, for example, can happen when a chromosome breaks due to chemical or even physical damage that happens. And that means that, there's a piece of DNA that needs to be repaired. Occasionally, that repair can happen through this sort of illegitimate
Mhm.
recombination process, and sometimes that leads to cancer. This was actually how that type of recombination was originally studied, is that people were investigating what happens to DNA in cancer cells, and they noticed that in some cases it's through this kind of illegitimate recombining that you can form genes that are, cancer causing.
Okay. Now, I have a question because yeah, that had never occurred to me before. I was reading your book. Illegitimate recombination, you say, happens a lot more than homologous recombination. If that in cells, right? This sort of random combining. But it occurred to me if that's a much more prevalent process, how come in sexual selection? How come it with eggs and sperm? How come they manage to do this beautiful homologous recombination and not not the other? What causes that?
Right. Well, let's just define when there are different situations when recombination occurs. What we're referring to in the book there that you're talking
Yeah.
about is the kind of DNA repair that happens after there's a double-stranded break
Yep.
in DNA, which is DNA damage.
Yeah.
And in that instance, the cell is very eager to repair that broken DNA. And so it's really just looking for a quick way to, fix those ends, seal them up, and allow the DNA to continue to be copied as DNA replication is happening and cells are dividing. And so, it's one of those things where there's kind of pressure in the cell to figure out how to quickly fix those broken ends. But what you're talking about uh, development, human development, and sort of egg fertilization and what happens at that stage of development is a process that's quite distinct because there we're not typically, at least, worried about DNA that's broken. We're actually just worried about DNA that is being very quickly copied and then is available to recombine using homologous recombination to make the kinds of recombined chromosomes that result from, from
So
sexual
so
selection,
when
and
it's
that's
when it's being copied, but it has to take parts of the chromosome from the father and part from the mother. So
mom and dad,
those
yep.
must be cut then, I would right? When, I guess at some
They
level.
do, but it's a much more controlled cutting process, I guess
Yeah.
is what I'm saying.
Okay.
Yeah.
Now, speaking of controlled cutting processes, which is what this is all about, CRISPR and your work and part of your work. The whole idea is it obviously is if you can edit the gene, that means you can determine the letters that are there and either correct mistakes or change it to make it better, and then we'll talk about all of that. But I was intrigued, you describe gene editing as 3 segments. 1, first recognizing the DNA sequence you wanna edit, then cutting it, and then, making and then you say the 3rd element is having an if having an editor which can be reprogrammed to look at different DNA segments. So and this is what obviously CRISPR eventually is all about. You have a reprogrammable device that can recognize DNA, go to that place and cut it. And that's the and you describe that. But the first thing occurred to me, in thinking about that is, why isn't there the 4th part, which is also putting add a DNA sequence part of that? Um, the cutting, what you so you go in and you cut, but then and we'll get to the fact that, as you say, cells like to repair and there's other ways to do it. But if you really want an editor, you've got to be able to make either add letters or add a sequence, and that's a central part of a gene editing. And I was surprised it sort of isn't thought of as a central part of that whole process.
Right. Well, I think it's because it's a little bit harder to do that as we discussed. So
Yeah.
what typically happens when DNA is broken in cells is that there's, this quick fix that occurs that is kind of this illegitimate recombination, if you will. And more rarely, more occasionally, there's a situation where there might be a piece of DNA that does have the kind of overlapping sequence, the homology that allows recombination that's more similar to kind of the normal recombining that we just discussed. And so I think that that's really the point of that explanation, was to point out that in many cases there simply isn't that piece of DNA available to do that kind of recombination.
Okay, yeah, no, it's interesting to see how that how the different therapeutic applications depend upon whether you can add something or just cut in 2 places and etcetera. I was gonna go into the precursors, this, ZFN and Talon, but I think given time the point is that, look, obviously, it's a holy grail, which has now been discovered, but a holy grail of, if you can obviously manipulate the human genome at will, you can do lots of wonderful things, and there were a bunch of tools that have been developed that have been used in a variety of things and were, in vogue around the time that you were thinking about something very different. You'd already moved to Yale. I noticed, I was a professor at Yale. You moved to Yale a year after I left Yale. I was looking at that, and then you
Which
had the
department were
wisdom to
you
leave
in?
and then you too had the wisdom to leave Yale. But anyway,
Which department were you in?
physics department,
Oh, physics. Yeah.
up on the hill there.
Yeah.
My dean was, Sid Altman, actually,
Oh, is that right?
my college dean, and I always thought, well, college deans, they don't do anything, and then after he stepped down, that's when they won the Nobel Prize. But, uh, anyway, you were studying RNA, and in fact, one of the first claims to fame of yours was to find the precise location of every atom in an RNA in a part of RNA that self-splices, which is the kind of thing that I guess Tom Cech had won the Nobel Prize and Altman had won the Nobel Prize for discovering that RNA can be chemically active as well. And so you were am I correct? You're the first person. Your lab was the first lab to understand that atomic structure directly and exactly, right?
That's right, and the motivation there was to understand the shape of the molecule, shape of the RNA that would allow it to work like a protein, even though it's not a protein. In fact, it's a very different chemical entity than a protein. And it's hard to kind of imagine now based on all the, research that's come since then, but in those days, people really argued about whether RNA would even have a three-dimensional shape associated with it. People thought maybe it's, more like a spaghetti molecule or something. I was convinced that it must have a particular shape to it that would allow it to function like a protein, except nobody knew what that would be. And so that was our motivation for trying to figure out the structures of these
And
RNAs.
what tools did you use for did you use to do that chemical structure
Well,
read?
primarily something called X-ray crystallography, where you literally form a crystal of the molecule that you want to visualize and then shine X-rays on it that are diffracted through the molecule, through the molecular structure in the crystal that can actually provide the data to back calculate the structure of the molecule. It's a really fun process.
Physics in action. In
Physics in
fact, it's
action.
poetic, of course, because it's
Yeah,
exactly X-ray crystallography that
yeah,
led your early hero, Jim Watson, and his colleagues
yeah.
to discover this the
And Rosalind Franklin, don't
and
forget
Rosalind
her.
Franklin, I said
Yeah,
call you, and Rosalind Franklin, her work, in fact, that they
right.
interpreted, to discover the structure of DNA. And
Yeah,
I think you just answered my question. I guess that wasn't clear to me. You said that RNA can fold up into three-dimensional structures was incredibly important, and I didn't get that, but now I do. It's that because proteins fold like that, and so it's that three-dimensional structure that makes it resemble a protein, and that's why it's incredibly important. Good.
that's right.
I didn't quite understand. Okay.
That's right, and, and sort of through the history of sort of modern biology has been driven in many, ways by understanding these kinds of molecular shapes, and in fact, I don't know if people paid attention to the Nobel Prize announcement this year, but in chemistry and in physics, actually, the Nobel Prizes were given out for artificial intelligence-based methods that
Yes.
allow, for the case of the chemistry prize, the prediction of protein
Yes.
structures.
Yeah,
Yeah.
no, yeah, and, um, and that's obviously protein structure is very important because that tells you, proteins do things, and if you can understand how their structure makes them do things, then you have a fundamental understanding, a chemical understanding, which is really what I guess has been your focus, and as a physicist, for me, the most interesting part of this is sort of the fundamental dynamics of how things work. Um,
Agreed.
but your discovery, your personal discovery of CRISPR, not CRISPR as a tool, but CRISPR itself, happened by serendipity. And uh, uh, um, a colleague who was looking at sort of Earth-based science, Earth and planetary science, looking at early history of life, and looking at the at something that was that and she was interested in you because you'd been looking she wanted to look at someone who knew about RNA interference. So which is, I understand the suppression of the expression of genes, the genes turning into proteins, and, and suppressing that as an immune response. You wanna walk me through that a little bit?
Yeah, wow, you're good. You have a lot of the details there.
Well,
Right, but
uh,
I'll fill in a few that you didn't mention.
I'm gonna take your
So
time. It seems I should do my homework too, so
well, no, 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 not at all.
listen.
This colleague of mine, Jillian Banfield here at Berkeley, was investigating bacteria that grow in the environment and the viruses that they coexist with. And she discovered examples of CRISPR systems, which are natural bacterial immune systems. This was before anybody knew that's what they did or anything about how they operated, except that it was pretty clear that these were somehow adaptable, meaning that they were mechanisms for cells, bacteria, to capture short pieces of DNA from infecting viruses and then make little copies of RNA of those viral DNA segments and somehow use them to protect cells from future viral infection. So it all sounded very mysterious, but also very interesting. And Jill did not, she's not an experimentalist herself, and she doesn't work on RNA, but she knew that I did. And so she reached out to me to ask if I would like to study this with her. And that's actually how we got introduced to both each other and to CRISPR.
Yeah, it's a wonderful serendipity. There's a few themes that I wanna come back to near the end, but one is curiosity-driven research. The other is science as a social activity. That it's this misc and physics is responsible for the misconception of because of Mr. Einstein that somehow it's due to someone sitting alone in a room at night discovering it, but that's not how science works. It's a deeply social process, and we'll get to that, but now I wanna talk about CRISPR itself, which is I mean, and it's a pity we can't show the diagram here, but it because that kind of makes everything clear. CRISPR stands for clustered, regularly spaced, interspersed, short palindromic repeats. And by the way, I have to say it's another reason I didn't go into biology. I couldn't remember all the names.
The acronyms, I
Yeah, the
know, it's
acronyms are
terrible,
just
right? Yeah.
yeah, but when you see the picture, it tells you what that means. So why don't you walk us through what that picture means?
Well, the acronym and the picture refer to this process that I just kind of mentioned briefly, which is that bacteria capture short pieces of DNA from viruses and they store them. They store them in their own chromosome. And they don't store them randomly, but they store them all in one place in the chromosome. It's like putting them all in a drawer of a bureau or something. And so in the case of CRISPR, these are places in the bacterial chromosome that have a whole series of these little stored pieces of DNA that are inserted between a sequence of DNA that is repeated over and over. So it's a very distinctive pattern. And it probably is there to help the cell know that, this is where I've got my, my immunity genes stored, and they are there to serve as templates for the production of molecules of RNA that are then able to find those viruses if they show up in the cell again, because as you mentioned earlier, RNA and DNA have very similar sets of letters and they can pair with each other and use that as a basis for recognition.
Yeah, yeah, I mean, in hindsight, things are always easy in hindsight. But when you see that and you see that the more of these, these unique DNA sequences that a bacteria has, the more it can fend off, viruses. I mean, it kind of jumps out, at least in retrospect, that, "Hey, this must be because it's been exposed to these viruses and it's learned how to build up immunity," and obviously something about that chemical composition allows it to have immunity. I mean, it just sort of stands out, and therefore that chemistry is somehow, um, it just in retrospect, when I was looking at it, hey, this is clearly the thing you should be looking at that's somehow going to be attacking, or manipulating RNA and DNA, but again, in retrospect, it's probably easier to see than.
And, there's a beauty to it as well, don't you think?
Yeah,
It's so elegant. It's such an elegant way to
unbelievably elegant, and
find viruses. It's
yeah,
really great.
and in fact, so, and the interesting thing is that these are present in archaea and bacteria, the oldest life forms on Earth. So the
Yeah.
immune system, so even these very, one could say primitive, I don't think that's the right word to use, but it is in a sense of at least temporally primitive, um, life forms, they have this remarkable chemistry and this immune system, isn't it? It's just, it is amazing.
It is amazing.
And
Yeah.
now the things that attack bacteria are bacteriophages, right?
That's right.
And they basically cut apart, the way they attack bacteria is they act like little scissors, and they basically go in, and they're viruses, and they cut apart the DNA of the bacteria and kill it. Is that okay?
Yeah, that's right. And they take over the machinery. They make more viruses.
And the process, just they're a virus, so
It's
they kill
so
it and take over the machinery and make more viruses that can go and kill other things. It's just,
more cells. Yeah.
again, it looks like, they're just, yeah, it's just amazing that they efficiently do what they need to do. One thing in your book that amazed me is that you said there are 10 to the 31 bacteriophages on Earth. And
Mind blowing. Yeah.
now, I don't know if anyone's ever, maybe they have, because you've talked about this a lot in different ways, but um, there are 10 to the 22 stars in the universe. So this is a billion times more stars than there are in the universe. It's amazing to think about that, isn't it?
Yeah. It's very, very, it's a very big number.
Yeah, I know. I mean, I try to put it in terms of, there's 100 billion stars in our galaxy, 100 billion galaxies, times 10 to the 22. This dwarfs that by a factor of a billion, and those are, and so they're the fundamental. It's obviously not only are they fundamental, but it was essential. If you think about the history of life, I guess, now that we're talking about it, it didn't occur to me till just now. Life had to develop an immune system if there are so many of these.
Oh, it was
We wouldn't be
essential. Yeah.
around today if we didn't
That's
have a
right.
way to handle that.
That's right.
And
That's
so
right.
it was a requirement, and undoubtedly, a lot of bacteria died until that requirement was developed by natural selection.
And by the way, some people think that, this is one of the drivers of evolution too, is that viruses are, they're a big pool. Think of all the genes in those 10 to the 31 phages, right? It's a lot. And so they are an incredible library of genetic material that, provides a resource for cells. So occasionally we know that actually maybe more than occasionally cells take up genes from viruses if they're useful. So
Yeah,
it's
in
a
fact, I mean, is it not true? I don't know if, isn't it true that perhaps a large part, I mean, the large part of our genome comes from eventually having built up from virus?
Definitely. Absolutely. That's true for humans, yeah.
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 Now, associated with this sequence, this clustered, regularly spaced, interspersed, short palindromic repeat, are other genes called Cas genes. And they're always nearby. And what's clear, in retrospect, I guess it had to be discovered by people, is that they're involved in slicing up, DNA and RNA, right?
That's right. Well, they encode proteins, right? So they encode proteins that, combine with the little pieces of RNA that are coming from these stored, viral segments. And together, that protein and RNA are able to search the cell looking for viral DNA sequences, latching onto them if they're found, and then cutting them up.
They and so this CRISPR DNA gets converted to by transcription, I forget,
Well, the DNA gets converted by
the
transcription to
transcription
RNA.
into CRISPR RNA.
Mhm.
And so
Yeah,
CRISPR RNA is the thing that seeks out the relevant DNA of the virus.
but not on its own. It has to do it with these Cas proteins.
Yeah, yeah, no, yeah, the Cas proteins, and apparently with another RNA, which we'll get to, which was essential part of your lab's work,
Right,
right?
right.
Um, um, and they basically pair together to seek out and ultimately seek out and destroy, seek and destroy, like a missile. it is an amazing. I mean, as you just said, it's an amazing defensive system, and it's remarkably complicated and elegant at the same time, and and um and yet it happens in bacteria. And,
Yeah.
when I have to when I thought of that, it reminded me, I don't know if you this quote from Bertrand Russell, who said, "From protozoa to man, there is nowhere a very wide gap in either structure or in behavior." And
So true.
yeah, I know, it's isn't
Well
it? And
said.
it's so true in so many ways.
So many
And,
ways.
and it's fascinating. Um, I guess I wanna jump ahead. I had notes later on, but the thing that really occurred to me is you're a chemist, you're a biochemist. This amazing mechanism, which we'll go into a little more detail, is chemistry in action. Life discovered this chemistry, bacteria discovered this elegant chemistry, and in some sense, I guess the interesting question is, scientists like yourself and other people had obviously gene editing is a is an amazing holy grail. It is interesting that you learned how to do it for bacteria. Do you see any way maybe with AI now? Do you think it could is there any way that you think a smart scientist would have said, "Oh, I can develop this molecular machine by putting these things together to do that," or did we have to learn from from life?
I think we had to learn from life, because I don't first of all, I'm not sure anyone would have had the idea for how to do it. And secondly, I'm not sure it would have been possible even if the idea, even if conceptually you could imagine something like this, how would you actually build it? And this has been one of the challenges in biology for forever really is, how do you construct molecules that have a particular property or behavior? Scientists have been trying to do this for all kinds of reasons, right? Whether it's that you want to understand just fundamentals about how life works versus maybe you want to be able to create molecules that do new things or are useful industrially, and yet it's really hard to do that. And the thing about cells and viruses and evolution is that they've had a lot of time,
A lot
right?
of
A
time, that's
lot more time than any of us have had. So
yeah,
they've had a
people
lot of time to do trial and error, and that's what they do. They basically do trial and error, and if something has gives cells or viruses an advantage in some way, meaning that it helps them to grow
yeah.
or replicate better, then those kinds of molecules and traits tend to get selected.
Yeah, no, I mean, that's the big feature of natural selection, which is so non-intuitive, and,
Yeah.
Richard Dawkins and I have talked about that on stage, so long with people who don't understand evolution, don't realize long time is something we just don't have we can't appreciate.
No.
It's just so non-intuitive. Billion mu not just thousands of years, but hundreds of thousands or millions or billions of years. It's just so
Yeah.
long. And so then necessity is the mother of invention. The fact is,
Exactly.
as you said, it's not
Yeah.
just this they were interested, what bacteria were interested in this, it's an essential tool of their
You
survival.
had to have it.
And
Yeah.
you could have imagined lots of failed trials, and those
Yeah.
life forms died out for that
Right.
very reason.
Yeah.
And it's just like natural selection, the only ones that survived were the ones that, through trial and error, managed to develop this incredibly amazing, sophisticated, tool and technology, but again, it was necessity, that
That's right.
and that does remind me of something that I was gonna ask about, a colleague, I was gonna say a friend, and we actually met at the World Economic Forum when I used to speak there, which I understand you spoke at. But, Francis Arnold won the Nobel Prize for basically, as a chemist, learning from evolution, right? Using directed kind of natural selection in her laboratory to help develop chemical processes that you might not have thought of in advance. It's an amazing tool if you can use it. And, um, now but the key thing was having seen these things, I mean, what your lab and what the goal was to learn exactly how CRISPR, RNA, and Cas what eventually would be called Cas9, but, how they seek out, how they pair, and, and do that. And so the there was another sort of bit of serendipity that led you to this. So, the serendipity of meeting someone who was interested in CRISPR and introduced you to it, and you knew you knew that you were working on RNA and had the technology to understand and try and explore those things. But there was this thing called type 1 Cas genes, and then there were these other which as far as I can tell, those Cas genes just ask like scissors and cut everything in sight. But there's another Cas gene called type 2 Cas gene, and there was another bit of serendipity that led you in that direction. You wanna talk about that for a second or a minute?
Yeah, I mean, I'm not there were a few,
Well,
I
I
guess
mean,
there were.
someone you met. I mean, again,
Okay,
you know.
let's start there. So, we we in my lab, we were working on these, CRISPR systems and trying to figure out their chemistry, how they worked, how they protected cells from viruses. And, it was a small project in my lab in those days, and I found myself, invited to meetings that I didn't typically attend. And one of them was a meeting being held by the American Society of Microbiology, and it was being held in Puerto Rico that year. This was in 2011. And so I had been invited to give a lecture there, and I'm not quite sure why I went. And I was kind of wondering at the time, is this really,
Indeed.
should I really spend time doing this? But it, I don't know, it was an interesting place
Yeah,
and
it's a
a
nice place to go, and
interesting topic.
yeah.
And I went to this conference and I ended up meeting a woman, Emmanuelle Charpentier. And Emmanuelle is a scientist who is I think she would define herself as a medical microbiologist. So she's very interested in bacteria that infect humans that cause, various kinds of, sometimes very severe or lethal infections. And in studying bacteria that can do this in humans, she had come across a different kind of CRISPR system. And so she was curious about how this might work and what it might have to do with the biology of this infectious bacterium. And so when we met at the conference, she asked me whether I would like to work with her lab to investigate the chemistry of a new kind of CRISPR protein, a Cas protein called Cas9. And I was intrigued. It sounded very interesting. Nobody had done much research on this particular form of CRISPR at the time. And so that was the really important, that was one of the really important things that happened in our careers because we were able to begin doing science together that probably neither of our labs would have been able to do independently.
Yeah, it was a symbiotic relationship, just like the
Yes.
mitochondria.
Well,
And, well, before I have some questions about that. Before I do, I wanted to I mean, the final step then, I mean, I wanna jump to the final step, which is the realization that your collaboration ultimately realized that in order to basically, um, produce CRISPR RNA in bacteria and attack DNA of phages, you needed 3 components. The CRISPR RNA, which was created by the CRISPR, from the CRISPR DNA, Cas9, which unlike the other Cas proteins, does a single cut. I guess, is that the key point about Cas9? Is it does
Well, it cuts DNA, and of course, DNA is a double-stranded
Yeah.
molecule, so it's making, it's cutting both of those strands. But I guess the important point is that it cuts once and it cuts precisely.
It cuts precisely, which is obviously position. And then the other aspect is that and people just tried CRISPR RNA and Cas9, it didn't seem to work. You discovered you need another bit of RNA, tracer RNA. And is the purpose of that tracer RNA to keep the Cas9 and the CRISPR RNA together? Is that what
Yes.
that's its purpose, basically?
Yeah, that's
Otherwise
fundamentally what it does.
otherwise the Cas9 isn't finding the CRISPR RNA. And so that's the key point, that it holds them together.
That's
Then
right.
when you do that, you can make a double-stranded cut, and then you're in business. And that basically, um, then you have a virus-seeking missile. And that was ultimately the work that you then wrote the paper, the science article, which was June 8th, I think, 2012. And that was,
Right.
I think that's I assume the paper in which both you and Emmanuelle won the Nobel Prize.
Yeah.
I mean, obviously did a lot of work after that, but now I do wanna ask about 2 aspects of that. I've already asked about Isn't it amazing that bacteria found the chemistry that science couldn't? But I think that's great because they're smarter than you think. And they also have behavior. That's the other thing that Bertrand Russell was talking about. And it's amazing. You can't tell from behavior. You can't tell about consciousness. That's because bacteria behaves, but it doesn't have consciousness. But what was it about? But I have two questions. W- What was it about your lab and your skills that caused Emmanuelle to want to work with you? What was it specifically that your lab could do?
Well, it's that we're biochemists and we work on RNA because, again, it comes back to the way that these virus-seeking missiles, as you just said, how they're guided. How are they guided? Well, they're guided by RNA, right? So if you want to understand how the missile works, you got to figure out the guidance system.
And did the but I given your history and understanding structural, I guess what we can call structural biology, I mean, the structure of molecules, was was that an essential part of understanding how the missiles guided? Did you do much structural work there, or was it experimental work on RNA directly? But
We did eventually, but initially in those very early months working together, we were focused more on biochemistry. It was really about testing how it was that these RNA guided proteins could find viral DNA and what they did when they got there, how they cut it up.
okay, now I want to get there are a bunch of morals I'm trying to get across here, and And by the way, I mean, you later on talk in the book about the importance of explaining to the public so the public can be informed about these things. And I mean, that's one of the reasons, that's one of the purposes of this podcast and the kind of things we're talking about is to try and give people the tools to understand what the basis of these things are so they can make informed decisions and equally importantly vote for people who might make informed decisions. But so that's why, that's why it's so interesting to talk about these details. But the the other aspect of this, which I think is so important, is that it was based on curiosity-driven research. It's it's you were you were asking fundamentals about RNA. Jill was asking questions about about uh, ancient bacteria. Um, and it wasn't as if people said, "Well, gee whiz, I'm going to take money to develop a gene editing scheme." Okay, and it's I like to and I you use some examples in your book, but I like to think the same thing. If you had paid Bell Labs in the 1940s or early 50s saying, "Make better computers," well, they would have had cogs and wheels, and it would have, but and then the transistor was invented, and the importance of curiosity-driven research is really something I wanna spend a few minutes talking about because of the times in which we live. Um, because of what seems to be such pressure to produce economic benefits, for science to be applied, and for governments to fund science that's applied. We see in the United States, I suspect, I'm not I'm not sure, but given the composition of the advisors to the new administration, I suspect we're going to see a lot more pressure to, look for economic benefits of science. In spite of the fact that years ago, I was involved, when we were trying to get governments to spend money on fundamental research, with a study that was eventually done for the National Academy of Sciences, uh, something before the gathering storm, it was called. But one of the things that was discovered was that, interestingly enough, half the GNP of the United States now could be traced back to curiosity-driven research a generation earlier. So I'd like you to talk about the importance of this a little bit because I think it's something when we think about one of the what may be some of the lessons that come from this podcast, that may be one. And I think the history of how this uh, unbelievably important application, a tool that can change medicine and biology, was not developed for that reason, or didn't wasn't discovered for that reason also. Sorry, but that's the pre
Yeah, no, it's a great point. I mean, yeah, and actually, this is the way that a lot of fundamental breakthroughs and
Mm,
discoveries have been made. They're somewhat serendipitous. They're due to scientists being curious about something. Think of penicillin as a great
yeah,
example of this, right?
absolutely, another one.
I saw another interesting statistic recently related to what you just mentioned about government-sponsored research, and that is that so but hopefully your listeners are aware. But if they're not, there's a major taxpayer-funded organizations in the United States known as the National Institutes of Health and the National Science Foundation.
Yes.
These are both government agencies that are responsible for distributing taxpayer money to people like me that are doing, fundamental research. So we write grants. This is the way we fund our laboratories. We write grants to these agencies and we propose projects and they get reviewed by our peers. Now, here's the statistic I saw recently. It turns out that for every dollar that the National Institutes of Health in the United States invests in that type of curiosity driven fundamental research, there's at least a $2.50 return in the economy later. And why is that? Well, it's because of what we just said is that a lot of discoveries come from working on fundamental questions in biology or in other areas of science and then making discoveries that have implications far beyond what the discoverer could have imagined. And another great example is the mRNA vaccine that was used during the pandemic. That science again came out of work that was initially very fundamental and wasn't designed to develop a vaccine per se, but in the end, when we needed it, we had the knowledge to do so, and we only had it because of that kind of funding.
Yeah, and you just remind me of another when a colleague of mine when I was at Harvard. I moved there from MIT, and an amazing scientist named Ed Purcell, who won the Nobel Prize for inventing NMR, which of course is
Mm.
relevant. But when he and Bloch invented NMR, nuclear magnetic resonance, um, he was asked about it and said it'll never have any applications. You couldn't, it was like it's a fascinating way of, understanding how nuclei work and how to manipulate them, but didn't realize those applications. And one of the reasons it's important, just yesterday, the day before, I was reading that the government of New Zealand has just do you hear this? They just they said we're gonna cut funding, and half of the funding of science has to now go directly to economic benefits. And it's the most misguided kind of action that a government could take, I think. And
Yeah, because the problem is it sounds like a great idea, doesn't it? But,
yeah,
the problem is which things are gonna be those things. We don't know.
exactly. You don't know till you know.
No.
And
Yeah,
that's what makes science so wonderful. You don't
right.
know till you know, and it's that thrill which you talk about in your book, and anyone who's done science is the thrill of exploring the unknown and you never know where it's gonna lead.
Yeah.
And another aspect of making sure these agencies, scientific agencies, which are ultimately directed by the public because governments determine the heads of those agencies, um, is that they spend money on science. One of the things that I've spent some time recently lately is that agencies like the NIH and NSF has devoted a lot of their energy instead of to science to social engineering, and and it's um and it's unfortunate, I think personally, because
Yeah.
there's so little dollars anyway. The NSF, I mean, NIH gets a lot more, but the NSF is maybe $8 billion, and it's just and you take $1 billion of that, or $1.5 billion of that, and spend it to trying to do social engineering that's probably more importantly done somewhere else, and you've lost a lot of the
It's
important
a problem.
science.
Yeah, I agree. Yeah. And
I
just a time check here because I'm gonna have to sign off in just about 10 minutes.
Okay,
So,
yeah, I'm not. I'm looking. We were 114, and I was thought it was go 130, but okay, we'll go 125. But okay, um, well, let's get. I want to talk about general trends then. I mean, okay, before we l- the other thing I do want to talk about, this led science, that discovery obviously changed your life as well as changing science, and it hasn't been the same since, as you talk about in the book at great length. But one of the things you did invent involved is creating companies. Everyone I know in your field seems to create companies, and and it's okay. That's good. There's a lot of possibilities. But I was going to ask, I when I was chair of the physics department, we created a program in physics entrepreneurship, and and we didn't know anything about that. And so one of the things we did was get physicists to become entrepreneurs to come back. And the one thing that they said we didn't teach. We wanted to find out what we should teach students. So we're doing a master's program. And they said, you got to teach people how to fail effectively. That's the one thing that, because we give full problem sets in undergraduate classes, we give graduate PhDs to, this is a problem that's likely solvable. But then you go into the real world and it's not done that way. And a lot of the times, in fact, the tools you develop for one thing, as your story is a perfect example of, ends up solving something that you had no idea. And you may have come up against a brick wall here. But so I wanted to ask, was it how much of it is a shock for you to become an entrepreneur or become what was your greatest learning curve or surprise in that regard as you went out of
Well,
academia to business? I mean, you didn't leave
well,
academia, but as you opened up that option.
yeah, yeah. I mean, I'm an unlikely business person in a way because it wasn't my goal at all in going into science or scientific research. But maybe we should just say a little bit about, why start a company?
Yeah,
Why do people do this? Are they
exactly.
just trying to get rich or what are they doing? And really, it's what's interesting is that we've been talking about how a lot of fundamental discoveries are made in academic labs. That's kind of what academic science is really all about. And, but as we also talked about, sometimes those discoveries have real world impact, and they have problem solving abilities, and they could be scaled, and they could have, they could change things in the world in positive ways. And yet academic groups for the most part are not they're not they're not set up to do that kind of scaling or development. And so that's really when I think companies make sense for science is when you have a discovery that has real world potential. It can do something and you can see it. You can see it being able to cure a genetic disease or change the microbes that are producing methane, which is a
Mm,
powerful greenhouse
yeah,
gas.
yeah.
And so the science behind that. It's just that in an academic setting, you don't have the resources to develop it into an actual tool that's going to solve those specific problems. And so then I think it really can make a lot of sense at that stage to found a company because what a company can do is to attract the people and the money and the space and the other resources that are necessary to do that kind of scaling. And so for me, in this whole process, I think that, when but way back, actually before we started working on the type of CRISPR that became the genome editing tool, we realized that a lot of the molecules that you find in these CRISPR immune systems are very useful. Bacteria use them,
Mhm.
but we realized no, scientists could use them for other things. Except that, again, as academics, we're not really set up to develop them in that way. So that really led to the initial founding of a company called Caribou Bioscience, which is a therapeutics focused company, and then later to founding other companies that would solve particular problems using CRISPR and CRISPR related technologies, but do it at a scale and at a level that would be difficult or maybe impossible to do in an academic lab.
Okay, but the question that's great. But beyond that, though, I mean, as an academic, as someone who's been brought up as an academic and a professor, one learns a certain skill set. Is it clear that or I mean, it's sometimes, I guess, a rude awakening. Presumably, the skill set to run a successful company, to be a CEO, is a very different skill set, or it's not always the same skill set. And so is it advisable or is it easy for scientists to make that transition? Was it easy for you to make that transition? I guess you did you ever were you ever the CEO of any of these companies, or did you just become an advisor?
No, never a CEO.
Good, okay, yeah, because
In
that's
fact,
a very skill set.
I tell some of our company CEOs that, I really admire their abilities as business people, because you're right, it's a completely different skill set in some ways. In some ways, it's not. I mean, I think just like a CEO, an academic research leader a professor has to lead a team. You have to, advise people. You have to set goals. You have to set deadlines. You have to raise money. You've got to produce something. In our case, our product is typically publications. And so those are, there's some parallels there. But I agree with you 100%. I don't think in academia we are not trained to have the kind of skills that you typically need to run a business.
Yeah. Yeah.
And
You have to be entrepreneurial. I remember
you have to,
our
yeah,
business
that's
school
right.
dean, when we created this master's program in physics entrepreneurship, said that was an oxymoron. But in fact, scientists have to raise money. They have to do all these entrepreneurial things. But
They do. The other
I'm
thing I've
worried about
noticed,
I'd
the
like
other
to
thing
spend more
I've
time on it, but I'm worried about time. I do wanna look,
yeah,
you've given short shrift to,
yeah,
obviously, people can imagine the implications of CRISPR, but the possibilities of are immense, and you spend a lot
yeah.
of your book talking about, I mean, in plants and agriculture and food and, potatoes, which I live in PEI here, are very important, and in actually, engineering animals and maybe in making designer animals, and then ultimately changing the human genome and in different ways. I mean, not just changing the genome, but but editing to help cure diseases, and that's happening, and that's the great excitement about specifically diseases where you know that there's a genetic cause, not that there's about 3,000 genes that cause it, but immunodeficiencies, Huntington's disease, sickle cell, cystic fibrosis, resistance to HIV, perhaps, muscular dystrophy, and maybe even cancer in some ways. So it's incredibly. I mean, I don't want to give short shrift to the. Obviously, that's why this work is so important. I want to ask 3 questions because I think we have 3 minutes left, about, if it's okay, and maybe 1 minute a question apiece. 1, Richard Feynman said, when he won the Nobel Prize, that that wasn't the exciting part. That it was the discovery that was more exciting. I Is that the way you feel?
Yes.
Mm,
In a word, yes. It's the process,
it's the process,
it's
yeah,
the whole
yeah.
experience.
Um, One thing, you spend a lot of the last part of the book talking about, this key question of affecting the human genome, and I was happy to see, because I totally disagreed with you. I don't see any real problems of doing research or curing disease. I think it's ultimately important, and sure, there are risks, and there's people are going to abuse it, but you don't have control of that over science. And it seemed to me that when you talk about your own evolution, it more or less came around to that. Um, um, but there is one thing that hit me when you said that CRISPR is made basically for $130, you can get a gene editing kit now. And I was a chairman of the board of the Bolton Atomic Scientists for over a decade that set the atomic clock. And we used to look at all the existential threats to humanity. And we got Matt Messelson, who you probably know, to talk to us once. This was 10, 15 years ago. And we were talking about bioterrorism. And he said, I'm not so worried, because you need, to do these labs for genetic, you really have to have an incredible lab. And it's not the kind of thing someone's going to have in their basement or whatever. But now one can do gene hacking in a basement. And so should we be more concerned about bioterrorism as a result?
No, I would say not. I still think you have to have a certain level of skill, and it's not something that is easy enough to do that that I would worry about it in particular relative to all the other things that people can get access to that can cause harm. So
Okay, well, when you were thinking about whether it's ethical, to affect humans, basically the embryo, and change the human genome, as you point out, when you talk to people who either had relatives who had a disease that might be cured, in some sense, the attitude is it ethical not to do that?
right.
Is it? And it reminded me of a more serious ethical problem, but it's that kind of problem. I remember when I was an undergraduate, I met someone who worked on the atomic bomb, a physicist, well-known person. I won't mention his name. And I said, "Do you regret working on the atomic bomb?" He said, "My brother was in the Pacific, and he and 2 million other people would have died if we had, I mean, if we hadn't developed that horrific weapon." So when it comes down to this, that it's not so clear or crystal clear. Um, the if you can save lives, is it ethical not to? And you can't control. No one can control the the results of their work, and no one can. And people say science is bad, but my friend Steven Pinker pointed out you could say architecture is bad because architects created the Auschwitz, and but it's, you can use science for good or bad. And I think I'll give you. I want to just read. I know we got to end, so I want to just read the last lines of your book. Okay. When it comes to CRISPR, the possibilities of this new technology, good and bad, are limited only by our imaginations. I firmly believe we can use it for the former and not the latter, but I'm also cognizant that this will require determination from us, individually and collectively. As a species, we've never done anything like this before, but then again, we've never had the tools to do it. And I thank you for helping create those tools, that, for better or worse, to potentially help our society. And I hope that our discussion has talked about, some of the aspects of science has been fascinating for me. I wish we had more time, but it's been a real pleasure. So I want to thank you for coming on, and I hope you enjoyed it.
Thank you for inviting me. What a great discussion. And like you said, lots to do. There's a whole future ahead for this. So everybody should stay tuned.
Yeah, stay tuned, and good luck to you and your colleagues. Thanks again.
Thank you so much.
Hi, it's Lawrence again. As the Origins podcast continues to reach millions of people around the world, I just wanted to say thank you. It's because of your support, whether you listen or watch, that we're able to help enrich the perspective of listeners by providing access to the people and ideas that are changing our understanding of ourselves and our world and driving the future of our society in the 21st century. If you enjoyed today's conversation, please consider leaving a review on Apple Podcasts or Spotify. You can also leave us private feedback on our website if you'd like to see any parts of the podcast improved. Finally, if you'd like to access ad-free and bonus content, become a paid subscriber at originsproject.org. This podcast is produced by the Origins Project Foundation as a non-profit effort committed to enhancing public literacy and engagement with the world by connecting science and culture. You can learn more about our events, our travel excursions, and ways to get involved at originsproject.org. Thank you.